Our today's topic is the ninth topic, and it is the general account of the Cordiatales. As you know, since last two video lectures, we are dealing with the fossil gymnosperms. So whenever we talk about the fossil, whatever the fossil record is there, we have to deal with that fossil record, and we have to take out the information, whatever the information is there, which is found in the fossil, and we have to study that information in order to conclude the which type of gymnosperm these are, and and to which type of the plant today's present day plants they show the affinity. That is, whatever the Benititles members are there, or whatever the Cordiatales are there, we have to judge their vegetative as well as the reproductive content, and we have to compare this vegetative and reproductive content with the today's gymnosperm in order to show their affinities. Okay, so today's topic is the general account of the Cordiatales. All right, now consider this. This is your syllabus. We are studying the paper 1 and the chapter 3, that is the phanerogams. Under this phanerogams, you have to study the gymnosperm as well as the angiosperms. Okay, we are here at the general accounts of the Cordiatales. Okay, we have completed the general accounts of the Cycadophilicales and the Benititales, and now we are going to see the Cordiatales. Remember, all these three are the extinct gymnosperms. Okay, all these three orders belongs to the extinct gymnosperm, and they shows the fossil forms only. And that is why, as I already told you, that there is a scarcity of the information whenever we talk about the fossil record. Okay, so let us consider the Cordiatales. Now consider the first introductory point. See, the Cordiatales, an extinct group of the Palaeozoic tall trees of the gymnosperm, form the world's first great forest. See, it has a very peculiar importance. See, whatever the Cordiatales members are there, whatever the members which found under these particular orders, they are all now extinct. Okay, they have all got extinct. They are all fossilized now. No present day member or no living member found in today's date. So they were the extinct group of the Palaeozoic tall trees. Palaeozoic means that literally means they have dominated the Mother Earth in the Palaeozoic era. So in the Palaeozoic era, there were the tall trees which are the typical gymnosperms, because they were showing the presence of the seed but not the fruit, and this makes them the gymnosperms. And these gymnosperms, which belongs to the Palaeozoic era, were the dominating trees. Tall trees were there, and these were the first plant to create the world's first great forest. Whatever the first great forest were created on the Mother Earth, this first great forest is created due to these members of the Cordiatales. See, the name was given in the honor of the Australian botanist A.J. Corda, because this scientist particularly has created the fossil record of this order, and that is why Cordiatales this name has given on the basis of the scientist name, which is called as the A.J. Corda. Okay. What are the Cordiatales? Cordiatales are nothing but the extinct group of the gymnosperm, okay, which were dominated the Mother Earth in the Palaeozoic era, and they were the first plant to create the first great forest of the Mother Earth. And this name is particularly given due to the scientist who has studied its fossil record and created this particular order, which is his name is A.J. Corda. Okay. Now consider the majority of the Cordiatales were the tall and the large leaves trees. How the trees are there? Trees are the large trees. Okay, they were the tall trees. Very, very tall trees with the very large leaves. Okay, leaves were very large, and their length is also very large. See, the trees were very tall, and they shows the large leaves, and which can attain the height of more than 30 meters. That literally means more than 30 meters. Literally means the 100 feet height is there for the plant. That is, every plant is showing the 100 feet height. You can observe in this particular picture. Such a type of the plants were there. This is the reconstruction of that plant, and these kinds of the plants were shown by the Cordiatales at the time of the Palaeozoic era. So if we were born in the Palaeozoic era, which type of the trees we are going to see? We are going to see the trees in the Palaeozoic era. This belongs to the Cordiatales. These Cordiatales were the quite a long trees, almost 100 feet in height, and they were showing the very large leaves. Okay, so large leaves were there, the tall tree is there, and they were making the world's first great forest, and they were dominated the Mother Earth in the Palaeozoic era. All right, consider this. See, the group started declining during the Permian period. Okay, in the Palaeozoic era, they have dominated the Earth, but in the Permian period, as the Permian period approaches, under this Permian period, it starts to decline. Okay, its number, whatever the number of members are there under the Cordiatales, they were slowly start to decline in the Permian period and become completely extinct by the end of this period. That is, by the end of the Permian period, all the members of the Cordiatales have become extinct. Now, why this happened? This can be happen due to the any reason. For example, the environmental change is the main, every time is the main reason for changing the flora on the mother Earth. Because whatever the Mother Earth, today we find whatever the flora is there on the Mother Earth, this can be changed due to the environmental changes. because at that time the humans were not dominating, so there is no question of the anthropogenic activity. Anthropogenic activities are not there at that time, but whatever the environmental changes are there, these environmental changes wiped out these particular members of the Cordiatales from the mother earth in the late Permian period. By the end of the Permian period, there were no Cordiatales on the mother earth. So literally from the Palaeozoic era to the end of the Permian period, till that time these plants were dominating the mother earth, but somehow due to the any environmental change, they have got extinct by the end of the Permian period. Now these Cordiatales occurred side by side with the Pteridospermials. Okay, we have studied the Pteridospermials, and these alongside with these Pteridospermials, these Cordiatales were dominating the mother earth. That is, their time period is quite matching. When the Cordiatales were dominating this mother earth, at the same time there were the presence of the Pteridospermials, okay, or the mother earth. So what is the reason of their extinction is not important, because any environmental change can happen there, and this environmental change can be responsible for wiping out this particular biodiversity of the Cordiatales. And these Cordiatales, you have to strictly remember that in the late Permian period they have got extinct, from the mother earth, and they were growing side by side with the Pteridospermials. Okay, now see the distribution where we have found their fossils. Okay, whatever the fossil remains are there, where we have found the fossil, depending upon that we can judge their distribution on the mother earth. Okay, now see the Cordiatales fossils have been reported from the North and the South America. That is the whole continent of the North and the South America. On both these continents we find the traces or the fossils of the Cordiatales. That is, these Cordiatales were typically found in the North and the South American continent. Plus, the Europe is also there. Europe is also occupied by these particular fossil record of the Cordiatales. That is, the Cordiatales members were also found in the Europe. After that, they were also found in the China as well. Okay, in the China region, you will also find the particular members of the Cordiatales, plus the Russia and also the India. That is, the Indian subcontinent is also showing the presence of the Cordiatales member, plus Australia and the Africa. Now you can imagine that the whole world is covered by these particular members of the Cordiatales, because these Cordiatales members are distributed all over the world. Now consider this: North America, South America, the whole Europe, the China, Russia, India, Australia, Africa. This literally covers the every continent on the Mother Earth. Okay. Now indicating that their worldwide occurrence during the Devonian and the Permian period. So by judging the fossil record, whatever the fossils we are finding today, depending upon that, you can clearly say that they were the cosmopolitan in distribution. Okay, they were found everywhere. They were cosmopolitan, right? So cosmopolitan distribution is there. They were found worldwide, particularly North America, South America, Europe, China, Russia, India, Australia, and Africa. In these places, we have already found out the fossil record. They may have present on the other places as well, but the fossil record, as we have discussed already, that it is a scarcity in the record of the fossils. Okay, because for whatever the fossils we have found so far, there will be the many more fossils which are yet to be discovered. Okay, but from these places you can conclude that they were worldwide in the distribution. All right, now consider this. See, the Cordiatales in India are represented in the form of impressions or the compressions of the leaves, seeds, and the petrified woods. That is, the leaves part, seed part, and the petrified wood parts were found in the India. Okay, in the Indian region you will find the impressions of the leaves or the seeds or the woods. Okay, and the members of the only family, that is the Cordiaitales, have been reported in the India, but the no members of the Paraxolaceae is found from the India, because these are particular order. These Cordiatales orders can be divided into the two families. The first family is called as the Cordaitales, and second family is called as the Porous Hyalure. Okay, so only the members of the Cordaitales is found from the India, but the no member of the Porous Hyalure is found from the India. So, whenever we talk about the Cordiatales and its Indian fossils, you should remember that the leaves, seeds, and the petrified woods have been reported from the Indian continent. Okay, so but the members of the only Cordiatales were found, but no Porous Hyalure members were found reported from the India. Now in India, it is represented in the lower Gondwana formation. Whatever the lower Gondwana region is there, this lower Gondwana region is generally showing the traces of the fossil record of the Cordiatales. You should carefully remember that whenever we talk about the lower Gondwana formation, they were found in the lower Gondwana region. Okay, and these lower Gondwana regions were typically showing the presence of the leaves, some form of seeds, and some form of petrified woods are there. Okay, so Cardiocarpus, then the Dodoxylon, then the Nigurithiopiopsis, and the Semeropsis are the Cordiaitalian genera reported from the India. Now see, whatever the names we are taking here, these are names are typically showing the genus or the parts which were found in the India. Okay, all these names, every time the fossil is found, for example, see, if there is a one plant and from this one particular plant only the leaves were reported, so these particular leaves has given some specific name. For example, cardiocorpus, these types of the names has been given to these particular parts. So whenever you see such a special kind of wording during the fossil record, they are indicating some kind of the plant part. For example, the leaves are there may be the root, there may be the stem, or there may be the seed, or there may be the some other parts of the plant, but these individual parts of the plant has given some specific name in order to study that particular part. Okay, so from India there were the many fossil records of the Cordiatales. The Cordaitales members were found, but not the Porous Hyalure member. And the lower Gondwana region particularly shows the presence of these Cordiatales members in India. Okay. Now see the distinguishing feature of the Cordiatales, whatever the features which are possessed by the typical Cordiatales. Now, whatever the features we are going to study here, scientists created these features by creating some imaginary plant. Okay. Now, we have the fossil record. And from this fossil record, if we connect all the fossils, for example, we have found the fossils of the leaves, fossils of the stem, then the fossil of the root. Now, once we have the fossils of all these three, we can construct the imaginary plant, that the roots may be like this. Then after that, the stem may be like this, it's a tall stem. Then the long leaves were like this. So we can construct the imaginary plant. And on the basis of this imaginary plant, we can conclude some of the distinguishing features of the Cordiatales. Now, from the UPSC mains examination point of view, this is very important because they will always ask you what are the distinguishing features of the Cordiatales. Okay. So whatever the distinguishing feature of the Cordiatales are there, we are going to discuss it now. Okay. Now consider the first one, that this group of the fossil plants had a tall trees with a slender trunk and the crown of the several well-developed branches. Okay. Now see what they first show. The very first thing which is clear about the Cordiatales, that they are the very tall plants. Okay, almost 100 feet. They are very tall plants. Now the stem is slender. Okay, like this. Stem is very slender, and at the apex they were showing the presence of the crown, like this. Presence of crown is there. Apex presence of crown is there, and this apex presence of crown is supported by the slender stem like this. So slender trunk is there, and the crown of the several branches were present at the apex. So whenever you talk about the Cordiatales, the first point you have to put forward is that they were the tall plants. Okay. The second thing you have to give is that they were showing the slender trunk. Okay. And in the third point, you have to show that this is a slender trunk possessing the crown of the leaves at the apex. So such a type of design is there for these particular plants which have dominated the Mother Earth in the Palaeozoic era. Now see the second characteristic, that these plants were present from Devonian to the Permian period of the Palaeozoic era. So you have to remember to give the period from where to where these plants have dominated the Mother Earth, right from the Devonian period to the Permian period. These plants were present on the Mother Earth. The leaves were quite simple. How the leaves were there? The leaves are simple. They are not compound. Okay. The simple leaf were there, and they are spirally arranged and strap shaped. Okay. How they are arranged? They were arranged spirally on the axis. So spiral arrangement is there, and these spiral arrangement leaves show the strap shape. Their shape is quite a strap-like, okay, and the grass-like or the paddle-like. So grass-like, as you can imagine, how the grasses leaf are there. They are just originating from the base and going like this. Okay, so this type of the leaves or the paddle-like leaves were there in these particular plants. So the long tree is there, the apex crown is there, the slender trunk is there, and the leaves are simple and spirally arranged while they were the strap shaped, and they sometimes looks like a grass-like or the paddle-like. Okay, but one thing is sure, that the leaves are quite long. Okay, they are lengthy leaves, and they are just showing the simple habit. Okay, they were not the compound leaves, but they are the simple leaves. All right, now consider. See, the leaves are attained the height up to the 1 meter. Consider this. 1 meter literally means the 3 feet almost. The 3 feet long leaves were there. You can imagine it is more than half of our height. Okay, generally the 6 feet man is there, and the 3 feet length of the leaves is attained by the members of the Cordiatales. So whatever the Cordiatales members are there, these are Cordiatales members showing the 3 feet long leaves. The leaves were just like the grass, okay, or even more sometime, and they had the parallel venation. Now this is very interesting, okay. Now parallel venation, generally you see parallel venation in the monocotyledons, okay, in the grasses, okay, because in the grasses you will see like this. See, the grass leaf is like this, and you will always see the veins are running parallel to the each other, okay, like this. They were showing the parallel venation like this. So this type of parallel venation which is found in the monocotyledons or the grasses, the similar type of the parallel venation is also found in the leaves of the Cordiatales. Whatever the members of the Cordiatales were there, the members of the Cordiatales showing the very long leaf, almost 3 feet or even more than that, and this 3 feet long leaf shows the presence of the parallel venation. Okay, now consider this. The scenty primary wood was present. Now this is very important. The scent containing primary wood is there. Okay, the stem, whatever the stem is created out of the wood, this wood is quite scenty wood. Okay, so scenty wood is there, and this scenty wood is supporting whole of the crown at the apex. Okay, so how the leaves were there? The leaves were quite 3 feet long, and they were showing the presence of parallel venation, and the scenty primary wood is also there. Then in the mature stem, the secondary wood is mostly pycnoxylic. Okay, as you know, monoxylic and pycnoxylic. In the last lecture, we have studied the monoxylic. It is the quite a porous wood which is found in the today's gymnosperm. But the pycnoxylic wood is quite a solid wood. Okay, it is very solid wood, and the secondary wood is giving strength to these particular stem. That is, these stems were having the secondary growth here, and they were creating the secondary wood. And this secondary wood is quite pycnoxylic in nature. That is, it is quite it has the quite solid formation. Now this is very obvious. If you have to manage the 100-feet plant, okay, then you must have the solid tree trunk. So this solid tree trunk is present in the form of secondary wood, which is mostly pycnoxylic. That is mostly in the very solid form. It is not the porous wood, but it's a very solid wood. Okay. So the sixth characteristic feature of the Cordiatales is that they were showing the presence of the secondary wood, which is quite solid and pycnoxylic in nature. Then they were showing the compound unisexual cones. Okay, that is the cones. Either the male cones were there or the female cones were there. That is, the unisexual cones were there. No bisexual cones were found in these particular plants. The members of the Cordiatales were always showing the unisexual cones. Either it will be the male cone on the plant, or there is a female cone on the plant, but not the bisexual cones. Okay, so unisexual cones were there. So these are all the very important characteristic features which are we have find out through the fossil record of the Cordiatales, that they were showing the 3 feet or the more length in their leaves. The leaves were showing the parallel venation just like the monocotyledons. Then they show the scenty primary wood. Then the stem also has the secondary wood which is pycnoxylic in nature, and the cones were the unisexual cones. Okay. Now consider some more. Each compound cone had a main axis. That is, whatever the cone, male cone or female cone is there, this male cone or female cone possesses the axis with the bracts sustaining the secondary fertile shoots. Okay, that is, they were showing the secondary fertile shoots above it, and it will constitute the whole cone. Either it's a male cone or the female cone. So below here we have the stalk, which is of sterile in nature, but above we have the fertile nature of the structure, which combinedly creating the male cone or the female cone. Okay, possessing either the fertile and the sterile appendages. So whatever the cone is there, each cone is possessing the axis, the main axis. Okay, the main axis is there with bracts. So whatever the modified leaves were there, which are called bracts, the bracts were sustaining secondary fertile shoots. So secondary fertile shoots were created out of it, and this whole structure can be considered as a cone. Okay. Now see, the megastrobili had a sterile appendages below and ovule bearing fertile appendages above. Now when we talk about the female strobili, what they are showing, the megastrobili is showing the sterile appendages below. See, consider this is a female cone, and this female cone at the below level, at this level, showing the sterile appendages. Okay, this area is showing the sterile appendages. That is, these are not fertile. Okay, this area is showing the sterile appendages, while the upper portion, this upper portion is showing the fertile appendages, and in these fertile appendages there will be the formation of the ovule. Okay, because we have found out its seed, and we can study where these particular ovules were present, and through these particular ovules through the study of this particular female structure, we can conclude that its upper portion, whatever the upper portion of the female strobila is there, it's the fertile portion, while the lower portion is the sterile portion, and the upper fertile portion is also holding the ovule. Okay, so how the megastrobili are there? The megastrobili are showing the presence of the lower sterile region, while the upper fertile region, and in the upper fertile region you will find an ovule. Okay, now see, 1 to 4 ovules were present in each female fertile appendages. See, very important. Not only the 1, 2, or 3, 1 to 4 ovules were actually present in these particular plants, which are showing the presence of these particular megastrobili or the female strobili. This female strobili is having the 1 to 4 ovules, and these 1 to 4 ovules must have get pollinated by the pollen grains from the male strobili, right? Now see the microstrobili or the male strobili. These male strobili had the sterile appendages below. They were also showing the structure like this, and the sterile appendages were present at the below, and the pollen sac containing fertile appendages above. Here you will have the pollen sac, and this pollen sac will contain the pollen grain, and this is the fertile region. So comparatively, whether it is the male cone or the female cone, basically the lower portion of the cone is the sterile region, while the upper portion of the cone is the fertile region. All right, so this type of the reproductive shoots were possessed by these particular plants. All right, now consider this. 4 to 6 terminal pollen sacs were present in each fertile appendages. Whatever the male fertile appendages were there, in these fertile appendages, 4 to 6 terminal pollen sacs were there. Okay, consider this: 4 to 6 terminal pollen sacs were present in the upper fertile region, and these fertile pollen sacs will create the pollen grains, and these pollen grains will travel through the wind to reach to the ovule in order to have the pollination, and after that the fertilization. So 4 to 6 terminal pollen sacs were present on the male fertile portions, while the sperms have not been reported, but the presence of the pollen chamber suggests that the motile sperm might have been formed. Now see, as we are dealing with the fossil record, okay, we do not have the fossil record relating with the sperms which are created out of the pollen grains. But these presence of the pollen chamber itself suggests that there will be the formation of the motile sperms. Okay, so even though we don't have the fossil record for these particular plants for their sperms, but still we know that once there is a pollen chamber, this pollen chamber must have produced the motile sperms which can fertilize the egg. Okay, and these motile sperm may have been formed there. Now consider this: the spawn. Now we have discussed all the general characteristics, or whatever the characteristics which makes these Cordaitales very different plants. These Cordaitales characteristics, all the 13 characteristics are of very important from the UPSC mains examination point of view, because these general characteristics or the distinguishing feature of the Cordaitales is most of the time is the favorite question of the UPSC mains examination. Okay, so all these 13 points you should repeat it very carefully. Okay, read it carefully and repeat it many times in order to understand it. All right, now see, these Cordiaitales are divided into the two families. Just we have discussed in the first few slides that there were the two families. Out of them, one family is reported from the India, while the another family is not reported from the India. Now the very first family, which is comes under these particular Cordiaitales order, which it is called as the Cordaitaceae, and the second family is called as the Porous Hyalure. Okay, so Cordaitaceae and the Porous Hyalure, these are the two families through which we can divide this particular order. Okay, so for example, see, now the many fossil records we have came across throughout the Cordiaitales members. Now some of the fossil record we are going to discuss here. Now all the fossils we are going to discuss, because maybe UPSC asks you some of the fossils. Even for example, the mesoxylon, they may ask you directly what is the mesoxylon. Okay, so that is why it is very important to study the whatever the fossil record we have regarding the Cordiaitales, we should discuss it carefully. Now see, under the family of the Cordaitaceae we have found the many fossil record. For example, the Cordiates. Now this name is given to the leaves, okay, because we have found the leaves of the leaves fossil of the Cordiatales, and the name for this leaves fossil is given as a Cordiates. Okay, then the mesoxylon. Now this mesoxylon fossil is the stem. It's the stem fossil of the Cordaitaceae family. Then we have the amyxylon, which is the root fossil of the Cordaitaceae. Okay, then we have the Quadianthus, which is the cones, okay, either male or female cones of the Cordaitaceae, and the Cordiocarpus, which is nothing but the seed of the Cordiaitailes. So whenever we talk about the seed fossil record, we are talking about the Cordiocarpus. Whenever we talk about the cones, you are talking about the Quadianthus. Whenever you talk about the root, you are talking about the Amizalon. Whenever you talk about the stem, you are talking about the Mesoxylon. And whenever you talk about the leaves, you are talking about the Cordiaitales. Okay. So you understand this. Whenever I say Cordiaitales, I mean I am talking about the fossil record of the leaves of the Cordaitales. Okay. While the second family is the Porous Hyalure, and this Porous Hyalure is showing the Porous Hyalon, which is nothing but the stem, and the Rhodospermum, which is nothing but the seed. All these fossil record we have with us, and it can be divided into two families, Cordaitales and the Porous Hyalure. And the one important point you already know about these particular two plants, that the Cordaitales members were found were reported from the India, but the members of the Porous Hyalure family is not reported from the India. Right. Now consider the very first family, which is called as the Cordaitales. Now see the general characteristics of this Cordaitales family. Now they grow luxuriantly, that is they also having the very good height and form the large forest of the tall trees during the upper Carboniferous period. Okay, that is the members of this the Cordaitales, where they were growing luxuriantly and they were actually creating the forest. Okay, that their numbers are so vast that they were creating the forest during the upper Carboniferous period. Now these plants have attained the height of more than 30 meters, as we have already discussed. That is nearly the 100-feet plants are there, and these Cordaitales members are attaining the very good height, almost up to the 100 feet. Now they had a terminal and spirally arranged and the well spread branches bearing the tuft of the leaves. Okay, so at the apex they were showing the presence of the branches like this, and these are branches showing the tuft of the leaves at the apex. Okay, so branching is there, tall height is there. For the Cordaitales family members, tall height is there, the branches are there, and these branches were creating the tuft of the leaves at their apex. Okay, now consider this. See, the leaves were quite large. Okay, we already know the members of the Cordaitales shows the very large leaves. Then the leaves can be leathery, they were grass-like or the paddle shape. So whatever the general characteristics which are possessed by the Cordaitales members, they were also possessed by the Cordaitales family. Hence these points are getting repeated. Okay, so the leaves are large, they were leathery, they were grass-like or they were the paddle-like. They can attain height up to the 1 meter. Yes, they can be up to the 3 feet, and the breadth of the can be the 15 centimeter. Okay, so 15 centimeter of breadth can be there, and 3 feet height can be there for the leaves. And the leaves, yes, they are the grass-like or the paddle shape. All right. Now consider they were, however, smaller than that of the cycads. Okay, they were smaller when compared with the cycads, while the some members also had the small needle-like leaves. While not all the plants were showing some large leaves like 3 feet leaves, but some members were also showing the needle-like leaves. Okay, so you carefully have to remember these exceptions, that there are plants which can show the presence of the 3 feet leaves, but there are certain plants which are also showing the presence of the needle-like leaves, needle-like small leaves. Now these leaves had the dichotomous venation. Now it is very important. Okay, because whenever we talk about the general Cordaitales, we say that there is the parallel venation, but there is a presence of the dichotomous venation. That is, dichotomy is also shown by these particular plants. So dichotomous venation or the dichotomy in their leaves were shown by the Cordaitales family members. This is the distinguishing point. Okay, so the plants were quite smaller when compared with the cycads, and they were sometimes showing the needle-like leaves, and they were showing the dichotomy in their leaves. Now, Greenland. Now, this is the name of the scientist. This Greenland scientist had studied and reconstructed the Cordaitalian plant. Okay, he had studied all the fossil record and he tried to create the imaginary plant out of it. According to him, that the plants have attained the height of the nearly 5 meters. Okay, some plants. Okay, some plants nearly attained the height of the 5 meters. That literally means they were the 15 feet in height. Not much more than that. That is generally the 30 feet is height. 30 feet range, 100 feet range is there for the plants in the Cordaitales members. But there are certain smaller plants as well which are not even matching with the cycads. Okay, so the height can attain up to the 5 meters with the steel root. Okay, as you know, what is the steel root? This is the basic botany. See, the steel root, these roots are those roots which are come out of the ground and support the plant, give the mechanical support to the plant, similar like the mangrove plant. Here I have put here the mangrove plant roots. Observe these roots. All these roots have come out of the soil and they were try to support the plant. Okay, so these type of roots are called as the steel root. Okay, and these are steel root, just like in the mangroves today. These similar type of the roots were found in the members of the Cordaitales. Okay. Now these studies suggest that the habitat of the Cordaitales is in the swamp along with the seashores. Now again, from this we can conclude that how they have evolved. Now if they were having the steel root, they may have present along the seashores, that is along the oceanic coastline. Whatever the oceans are there, along the coastline of these oceans, they were growing themselves, and that is why they were showing the presence of steel root. Because where we find today, where we find the steel root or the nematophores, we find along the seashores. Whatever the mangrove plants which are growing along the seashores, they were showing the presence of steel root, or those roots which are come out of the ground and support the plant. So from this point we can conclude that the members of the Cordaitales were also showing the presence of the steel root, or they may also showing the similar type of habitat just like today's mangrove plants. Okay. Now anatomy of the Cordaitales, whenever we talk about the anatomy of the Cordaitales, we have the stem record of the Cordaitales. The stem is resemble closely with the conifers. Okay, this picture, this is not the fossilized record, but this picture belongs to the conifers. Okay, whatever the today's conifers are there, these are showing the stem like this. Okay, so similar stem like this is shown by the Cordaitales member. Both the Cordaitales and the mesoxylon possess the large central pith and the cortex. So large central pith is there, then there is a much more cortex surrounding it. Okay, so the characteristic feature anatomically possessed by these particular members of the Cordaitales family, they were showing the stem similar like the conifers. Central pith, large central pith is there, which is surrounded by the cortex. Then the wood was scenty. Yes, there are some woods which are showing the scent in their species, in their stems, right? In some species, while the others develop the large vascular cylinder. Okay, large vascular cylinder is created, and in still other cases, the distinct growth rings were present. So diversity is there. Okay, so some wood is showing the scenty wood, primary scenty wood, simple wood. While the others were showing the presence of the large vascular cylinder, that means the secondary growth is there like this. The very thick stem is there and the secondary growth is there, and sometimes the growth rings were also present, which is showing the presence of the secondary growth. Okay, so right from the simple primary wood to the secondary wood, this variation can be occur in the members of the Cordaitales. That is, the Cordaitales members were not only showing the simple primary wood, but they also showing the complex secondary wood as well. So variation is there from species to species, because as there are the many species, we don't know the exact number at the time, what number they possess at the time of the Permian time or the Palaeozoic time, which for example we cannot conclude that there are the 100 species or there are the 1,000 species, right? But these species can range in their woods. Okay, they can show the primary wood, they can show the secondary wood. Okay, but anatomically they were similar with that of the conifers, in that they were showing the presence of the central pith and there is a presence of the cortex. Okay, now consider this. See the roots, how the roots were there. Now the roots of the Cordaitales are known as the amelion, and this resembles very much like the modern conifers. Okay, whatever the conifer plants are there, these are conifer plants, whatever the roots are shown by these are conifer plants, similar type of the roots were also shown by the Cordiatales. Okay, now consider this. The Greenland again studied the root system of the amelion and found to be that it is shallow and highly branched, forming the steel root supporting the stem. Again, consider this. This is nothing but the, just a minute. This is nothing but the steel root. Again, like the mangrove plants. Okay, so the members were showing the steel root system just like the mangrove plants, and they were quite the shallow. That is, they were coming above the ground and suggesting that their habit must be around the seashores. Right. So we, according to the Greenland, again the roots are of roots are creating the branching, and they were shallow. That is, they were present above the ground. Right. Now consider this. They were diarch to tire in structure, and ectotrophic mycorrhizal fungi were also present. See, very important, the ectotrophic mycorrhizae, that is endotrophic mycorrhizae and ectotrophic mycorrhizae. We have studied both type of mycorrhizae, and these ectotrophic and the ectotrophic mycorrhizae were also present in the members of the Cordaitales, and they were maybe the diarch and tire in structures. Okay. Okay. Now see, we are talking about the roots of the Cordaitales. Now see, anatomically, the roots of the Cordaitales members, the protoxylem had the spiral tracheids. Okay, whatever the protoxylem is there, it is showing the presence of the spiral tracheids, while its metaxylem was scalariform. Okay, scalariform in structure is present in the metaxylem. That is, whatever the protoxylem is there, whatever the metaxylem is there, these are the two kinds of the xylem depending upon when they have originated. Okay, the protoxylem shows the presence of the spiral tracheids, while the metaxylem is the scalariform. These tracheids had the multi-serried border pits. Okay, very important. Whatever the tracheids are there, these tracheids were showing the border pits. That is, these are tracheid elements showing the pits in their borders like this. Okay, so which is quite similar like the today's some of the plants which are showing the border pits in the tracheids. So the protoxylem is showing the spiral tracheids, while the metaxylem is show the scalariform structure. These tracheids were also showing the presence of the border pits in their structures. While the cortex was quite large, just like we found today, the cortex were present in a very large quantity in today's gymnosperms as well. If you cut the today's gymnosperm root and observe it under the microscope, you will again find the large amount of cortex is present there. So on the similar line, the cortex in the members of the Cordaitales were also large, and it can be divided into the outer and the inner cortex. That is, the outer cortex is there and the inner cortex is there. So the cortex is present in the large quantity in the members of the Cordaitales, and this large cortex can be divided into the outer cortex and the inner cortex. While the secondary cortex and the cambium were also quite distinct. That is, after the formation of after the secondary growth, whatever the cortex is created, this cortex is called as the secondary cortex, and the cambium is the tissue which is mainly responsible for giving the secondary growth in the plants. These cambium cells can grow rapidly, and they can create the new cells in order to increase the girth of the roots. And due to this cambium tissue, the girth of the root is significantly increased, and this will result in the formation of secondary growth. And in this secondary growth, there is a formation of the secondary cortex. So primary cortex and the secondary cortex, both cortex are there. Primary cortex are quite large, and they can be divided into the outer cortex and the inner cortex, while secondary cortex and the cambium is also very distinct, which can be found out in the fossil record. Now consider the leaf. How the leaf of the Cordaitales leaf, which type of the characteristics were possessed by the leaf of the Cordaitales. See, the Cordaitalian leaf is described under the name of the cordiates. Okay, this name is given to the fossil record of the leaf. Now the several xerophytic internal characters were present in the leaf. Now this is very very important. Okay, that is, the leaves is showing the xerophytic characters. Xerophytic characters are those characters which are possessed by the desert plant whenever there is a scarcity of water. If the water is not available in a plentiful, the plants were tend to have the xerophytic characteristics in their leaves, because they try to reduce the surface area of the leaves. For example, if the normal leaves are like this, the xerophyte leaves are like this. They were showing the needle-like leaves. Why? Because in order to reduce the water loss through the process of transpiration. Okay, this is very important that we know the Cordaitales family members is showing the xerophytic internal characteristics in their leaf. That from this point we can conclude that there must be the water scarcity at that time. During the Palaeozoic era, whenever these Cordiatales members are growing there, or the Cordaitales members are growing there, these members are facing the scarcity of water. There will not be availability of water, or the plentiful water is not present there, and that is why they were showing the xerophytic internal characteristics in their leaves. Okay, now the leaves are showing the epidermal and the hypodermal cells, where the thick walled and the hypodermal cells on the both the sides, and were grouped into the ribs. Okay, so epidermal and the hypodermal cells were showing the thick walled cell walls. That is, their cell walls are quite thick. This is showing how they are showing the xerophytic characteristic. And the hypodermal cells on the both the sides were grouped into the ribs. Okay, they can group into the ribs on the both the sides. Which cells? Only the hypodermal cells. Okay, not the epidermal cells. While the epidermal and hypodermal, both the type of the cells which are present in the leaf, they were showing the thick walled cells, showing their xerophytic characteristic. Several mesorge vascular bundles were also present. Whatever the vascular bundles were present in the leaves for the supply of water and the food, they were of mesorge kind. Okay, mesorge kind of vascular bundle were present in these leaves. Now each vascular bundle is surrounded by the thick walled strong bundle sheath. Okay, so if there is a vascular bundle which is made up of the xylem and the phloem, and this xylem and phloem, which collectively called as the vascular bundle, this vascular bundle is covered with the thick strong bundle sheath. Okay, bundle sheath is also surrounding these particular vascular bundle. Now then, the transfusion tissue were present in the form of some of the elongated cells between the two vascular bundles. Okay, if there are two vascular bundles like this, and the first vascular bundle is covered with the strong bundle sheath, and the second is also covered with the strong bundle sheath, and whatever the transfusion tissue is present between them, these are transfusion tissue were present to form the elongated cells. Okay, elongated cells and the transfusion tissue were there. Okay, so transfusion and the elongated tissue, whatever the transfusion cells were there, they were creating the very elongated cells like this. between the two vascular bundles. Okay, so how the vascular bundles are there in the leaf? The vascular bundles are covered with the strong bundle sheath, and between the first vascular bundle and the second vascular bundle, there is a formation of elongated cells. What type of cell is present there? Elongated type of cells were present there, and these tissues were called as the transfusion tissue. Now the mesophils were clearly differentiated into the palisade and the spongy parenchyma. Just like today we find in some of the angiosperms that the mesophil tissue is divided into the two, that is the palisade tissue and the spongy parenchyma tissue in some species of the Cordaitales, for example Cordiaitales lingulatus. Okay, so the differentiation in the mesophily can also be seen. So you can say that these were quite of the advanced plant, and they were showing some of the advanced characteristics as well, just like that of the angiosperms. So mesophil tissue is divided into palisade parenchyma as well as the spongy parenchyma. Okay, now let us consider the spore producing organs. Which type of the spore producing organs which we have found in the fossil record for the Cordaitales or the Cordiatales in and all. See, the strobili were usually monoecious, that is, it will be either male strobili is there or the female, but there are some members which are showing the dioecious nature as well, but never the bisporangiate. That is, the male strobili and the female strobili is never together. Okay, so they were always distinct. Either they are showing the monoecious condition or the dioecious condition, but the bisporangiate condition never found in these plants. Okay, so very important that you should remember that, because we have studied the bisporangiate condition in the Gametophyte. Okay, so you should carefully remember that the sometimes in the fossil record for the some another order we have find out that there were the bisporangiate cones, but in the some orders, for example Cordiatales, you will never find the bisporangiate condition. Either there is a monoecious condition or the dioecious condition, that is either the male and the female gametophyte is present on the same plant or on the different plant. That is, they may be monoecious or the dioecious, but they never came together in the single cone. That is, they were never bisporangiate. Okay, now the fructification were born on the slender branches of the 10 centimeter in length. Okay, whatever the male cone or the female cone is there, the 10 centimeter length is attained by these cones. These branches develop on the stem along the leaves. Okay, normally whatever the stem is there, these reproductive branches were also born on these particular stem, and these particular stem were also showing the presence of the stem, also presence of the reproductive branches, and these reproductive branches can be of 10 centimeter in length. Okay, now the slender stalk had the many stiff and the tapering bracts. Bracts are nothing but the modified leaves for the reproductive structures. Okay, now consider, we already know about the general characteristics of the Cordaitales. This is the long shoot and this is the complex apex. Okay, long shoot and complex apex is there, and this complex apex is showing the presence of 10 centimeter reproductive shoot like this, and the leaves are getting converted into the bracts like this. Okay, and they were creating the cone. Okay, the cone were created out of it, and the cones were either the male cone and the female cone. They may be present on the same plant or the different plant, that is the monoecious or the dioecious, but never bisporangiate. That is, never they come in the same cone. That is, the male and female organ are never present on the same cone. Okay, now consider this. See, a short bird-like strobili were present within the axilla of each bract. Okay, bract is nothing but the modified leaves. For example, see, this is the bract, which is the modified leaf, and under this bract, in the axilla of this bract, you will find the bird-like strobili like this. The strobili were born like this in the axilla of the bract. Okay, now the bracts were probably spirally arranged. How they were arranged? Probably they were arranged spirally. Okay, spiral arrangement must be there, and this spiral arrangement is showing the presence of the male or the female reproductive structure. Now each strobili has attained the length of the 1 centimeter. 1 centimeter length is attained by these particular strobili. Okay, so lengthwise the reproductive shoot can be the 10 centimeter long, and the cones can be the cone birds can be the 1 centimeter long. Okay, we are talking about the bird-like structure which is called as the strobili, and it can be the 1 centimeter long, while the reproductive shoot can be 10 centimeter long, which can contain the modified leaves which are called as the bracts. And in the axilla of this bract, you will find the presence of the 1 centimeter bud. Okay, now see, both the male and female reproductive organs are known as the Quadianthus. Okay, which name is given to this male and female reproductive organs or the male and female reproductive cones? It is called as the Cordianthus. Okay, now consider this. See the male strobili. Let us first consider the male strobili. How the male strobili is present in the Cordiatales. See, it is consisted of the thick central axis possessing the many spirally arranged bracts and some microsporophylls. See, The central solid central axis is there for like this, and then there are arranged spirally arranged bracts on it. Okay, like this. Spirally arranged bracts are there, and the central solid axis is also there like this. This is the general appearance of the male cone. Okay, and some of the while there were the some there are the bracts, for example they are the modified leaves, and out of them some are the microsporophylls which are showing the presence of the male gametophyte. Okay, some are just the modified bracts and some are the microsporophylls. Now at the tip of each microsporophyll there were the 1 to 4 microsporangia at the tip here. You will find, for example, this is the microsporophyll, and here at the tip of the microsporophyll, you will possess the 1 to 4 microsporangia. These sporangia were probably dehiscence longitudinally. Okay, longitudinally it will break out and it will give to the male, its surrounding the male gametophyte. Whatever the male gametophyte, that is the pollen grains were created there, they were longitudinally dehiscence from these particular microsporangia. So how the male strobila is there? Male strobila is nothing but it is consist of the two structures. Central axis is there, then the spirally arranged leaves are there. Okay, like this, and some of these leaves are the microsporophylls. At the top of these microsporophylls you will find 1 to 4 microsporangia, and these are 1 to 4 microsporangia. When dehiscence it will give out the pollen grains longitudinally. All right, now consider this. The microsporangium wall was probably one cell thick. Okay, whatever the wall is there for the microsporangium, it is one cell thick only. Okay, one cell thickness is present in the wall of the microsporangium and enclose many microspores. That is, the many microspore literally means the pollen grains. Many pollen grains were present inside this microsporangium. Okay, the pollen grains may be elate or range from the monolate to the triolate. Okay, now these are all the types of the pollen grains. We are going to study these types of the pollen grains in the angiosperm section. Here we have to remember that they may be the elate type or they can range from the monolate to the triolate. Okay, so whatever the pollen grains are there, they may be monolate to triolate or they may be elate sometimes. And these pollen grains can dehiscence longitudinally from this microsporangia to go into the air and come in contact with the female strobili. So whatever the microsporangium is there, its outer wall is only the one cell in thickness, while in the center it will have the microspores. Okay, now consider this. They are the monosaccate with the sacs attached on the both distal and the proximal poles. Okay, monosaccate condition is there, that is only the one sac is there, and this one sac is present at the apex as well. It is attached to the apex of the microsporangium as well as the base of the microsporangium. So the monosaccate or the only one saxus is there, and this one saxus is harboring all the male gametes there, that is the microspores. Now let us consider the female strobili. Okay, which type of the female strobili is possessed by the members of the Cordiaitales? See, similar to the male strobili, the female strobili also had a stout axis bearing the large number of spirally arranged bracts. Similar arrangement is there. What is there? There is a central bract, and on this there is a central strong stout axis. Stout means strong. Okay, strong solid axis is there, and it will possess the spirally arranged bract like this. Okay. So the general appearance of the male and the female cone is quite similar. The male cone is also possessing the central axis and the spirally arranged bract. So does the female strobili, which is showing the presence of the central axis and the spirally arranged bract. So the general appearance either of the male strobili or the female strobili is similar. Okay, the bracts were more in number than the male strobili. Okay, what is the difference? These number of bracts were more as compared to the male cone. Okay, when we compared with the male cone, you will find the more number of bracts in the female cone when it compared with the male cone, right? Now see, the two or more ovules were present at the apex of each megasporophyll. So out of these, some are the megasporophylls, and at the apex of this megasporophyll you will find 1 to 2 ovules were present in the megasporophyll. So 1 to 2 ovules were present in this megasporophyll, and we have discussed the male strobili, and these male strobili, when they were released, these pollen grains definitely has to reach to these ovules in order to pollinate and fertilize these particular female strobili, right? So how the female strobili is there? The male and female strobili general appearance is same, that is there is a central axis and there is a presence of the spirally arranged bracts, but the number of bracts were more in the female strobili as compared to the male strobili. Okay, and at the apex of each megasporophyll you will find 1 to 2 ovules. Okay, Now consider this. In the Cordianthus williamsoni, okay, this name is given to this particular plant in which this particular female reproductive structure has been found. A single ovule was present in each of fertile appendage. Okay, that is the each of fertile appendage. Whatever the megasporophylls are there, these are megasporophylls at the apex, showing the presence of one ovule, and the probable fossilar seed for these particular Cordaitales member is drawn here. How the seed is there, and from this seed you will have some idea about the ovule, which type of the ovule is possessed by these fertile branches. Okay, see, the ovule was the biatigmic, and the integuments were free in the lower part but fused at the apex. See, integuments were present there, just like the angiosperm and the today gymnosperm. You will find the outer on the outer side, inside you will have the ovule like this, and on the outer side you will find the presence of the integuments, that is the protective covering to these particular ovules. These protective coverings are called as the integuments, and they are biatigmic, that is the two integuments were there, and these two integuments were fused above, that is they were fused here but they were free at the base. Okay, so this type of structure were found in the ovule of the Cordaitales member or the Cordaitaceae members. Whatever the Cordaitaceae fossil record we have found out, in that we have found out that there is a presence of the ovule, central ovule is there, and this central ovule is biatigmatic, that is showing the presence of two integuments, and these are two integuments is showing the presence of the protective covering, and these two integuments were fused at the apex but they were free at the base. Okay, now see, the nucellus of the ovule was free from the integuments throughout. See, whatever the nucellus is the mass of the tissue which is present inside the ovule. Okay, so this is called as the nucellus, and this nucellus is free from the integuments. Okay, it does not come in contact with the integuments, but it is present at the center like this. Here you will find the presence of the integuments, and the prominent beak with the pollen chamber was also present. Okay, so the prominent beak is also present, which act as a pollen chamber later during the pollination and the fertilization. So see, here you can see the nuclear beak here. This portion is called as the nuclear beak. Here we will have the megaspore or the ovule structure, and this ovule structure is showing the presence of the nucellus, which is free from the integuments. Okay, it is not attached to the integuments, and on the other side you will find the presence of the integuments here. Okay, like this. Then see some more about it. See how the male gametophyte is there. Finally, how the pollen grains were created there. See, much is not known about the male gametophyte. Much is not known about the male gametophyte of the Cordaitacean members. Some only the upper part of the nucellus provides some picture of the male gametophyte. So whatever the fossil records are there, these are fossil records is showing the presence of the nucellus, and this nucellus which is present in the ovule in the female gametophyte is showing the presence of the pollen chamber, and this can be from this we can conclude that the pollen grains were created finally in the male gametophyte, and these pollen grains are received in the pollen chamber. So the male gametophyte, although we do not know much about it, but we definitely know there is a creation of the pollen grains, because in the female gametophyte you will see the pollen chamber. Okay, only the upper part of the nucellus provides some of the picture relating with the male gametophyte. Now see, the inner structure was the multicellular mature pollen grain were present in the micropylar canal. Okay, so whatever the fossil record we have find in the micropylar region of the ovule, we have found some of the pollen grains. So from this it is clear that from the male gametophyte, from the male cone, the pollen grains are traveling right to the sender, right to the micropylar end of the ovule, and from this micropylar end the pollination and fertilization is occurring. This we know about the male gametophyte regarding the Cordiaitales. Okay, now consider this. It could however not ascertain that whether the cells are multicellular region were vegetative or the spermatogonous in nature. That is, whatever the multicellular region is there, whether it's a vegetative region or the spermatogonous region, that is which will give rise to the spermatogonia or the male gametophyte, it is not clear, because the whatever the fossil records are there, we do not have up-to-date fossil record regarding the origin, whether it's a vegetative origin or the spermatogenous origin of this particular multicellular region. Right? Most probably, we can say that both vegetative and the spermatogonous tissue were present in the pollen grains. Whatever the pollen pollen grains were created there, these pollen grains may have some vegetative as well as the spermatogenous region in them. Because whenever we talk about today's pollen grain, what we do, we create the structure like this. The inner layer and there is outer layer. Inner layer is intine, outer layer is exine. Outer layer is generally spiny, and some of the cells inside here will create the sperms, and some will remain as it is. So at that time, whatever the pollen grains are there in the Cordiatales, these pollen grains must be showing the vegetative portion, some of the spermatogenous portion. The vegetative portion maybe remain as it is to provide nutrition, while the spermatogenous region may have created the sperms in the pollen grains, right? This could be okay, because we do not have definite evidence regarding this. All right. Now see the female gametophyte or the embryo. How the female gametophyte or the embryo is finally created. See, not much is known about this structure as well. That is the female gametophyte and the embryo. Some seeds with well-preserved female gametophyte in the cardiocarpus has been found. So cardiocarpus, this name is given to this female gametophyte. The certain fossil record we have with us, and depending upon the certain fossil record, we have created this particular picture. Okay, here whatever the picture you are seeing, you have to draw this particular picture in your home assignment as well, because this type of the structures may have developed in the seeds or in the ovules in the members of the Cordiatales. Okay. Now consider this. In a few specimens, they have also preserved the archaeogonia. Even the archaeogonia were found in some of the fossils. Okay, inside the ovules, they have found the archaeogonia. That is the egg. Okay, which is fertilized by the sperms, which are liberated by the pollen grains. The elongated gametophyte in such a specimens had only two archaeogonia. Okay, that is the two archaeogonia were found in these particular specimens, each of which exhibit the beak with a projection like the endosperm, projection of the endosperm. So each of which is possessing the beak in their structure, and they were showing the projections for the endosperms as well. Okay, endosperm projection is there, and the beak-like structure it is present in the archaeogonia. So the two archaeogonia are present inside the female gametophyte. Now, and check this seed, how the seed is got created out of this. Once, see, this is the micropylar end, right? If you consider this as a ovule, okay, for example, consider for a second this is an ovule, okay, and forget about rest of the structure. This rest of the structure is developed later once the fertilization occurs. So through this micropylar end, the pollen grains may have entered inside, and here they must have found the two archaeogonia, and once the pollen grains has liberated the sperms, they may have fused with this and fertilization occur. And once the fertilization is occurred, the embryo is got created, and then this embryo is got converted into this whole seed. Okay, and this is the probable fossilar seed of the Cordiatales. Okay, this structure, this whole structure is the probable seed which is created after the fertilization. Now see how the seed is there. It is believed that the Cordianthus type of the strobili were possess the seeds known as the Cordiocarpus. Now see, Microsporangium and the Camerospermum are the two seeds of the genera Cordaitales. That is, these are two structures were found which are relating with the seed in the fossil record. The names are given such as the Microsporangium and the Camerospermum. Okay. Now seeds were basically heart shape. Which type of shape is there? The heart shaped seeds were there. You can understand this is an heart shaped seed is there, and seeds are generally showing the shape like this. Okay, like this, and they were bilaterally symmetrical and crassinucleate. So seeds were heart shape and they were bilaterally symmetrical. That is, if you cut it from the center, you will exactly have the two parts. Okay, so that is why we are rightfully explained them as a bilaterally symmetrical, and they were crassinucleate in nature. Now see the central nucellus. Whatever the central nucellus is there is surrounded by the two layer envelope, of which outer layer was probably expanded to form as a wing. See, the nucellus is present at the center and it is covered with the outer layers, while the two outer layers were there which are protecting this particular seed structure, while the outermost layer is got converted in the form of wings like this. See, wing-like structure is there, and this wing-like structure, as you know, why this is important? It is very important during the dispersal of the seed. Whenever the seed tries to disperse from one place to the another place, this dispersal is very important, and for this dispersal they have created the wing-like outer covering. Okay, what it is called? It is called as the wing, which is forming the outer covering. All right. Now see, The Cordocorpus, in the Cordocorpus spinanthus, the seeds were large and it is surrounded by the 5 distinct layers. Okay, so 2 to 5 distinct layers were there. It will depend on which type of seeds we are talking about, because many seeds have been unearthed today, and we have examined them, and 2 to 5 layers may be there. So in the Cordocorpus spinanthus, you will find the 5 distinct layers are there in the seed structures, including the 2 layers each of the sarcotesta and the sclerotesta, and a layer of the endotesta. That is the 2 layers which are called as the sarcotesta, 2 layers which are called as the sclerotesta, and the 1 layer which is called as the endotesta. That is 2 plus 2 plus 1. That is the equals to 5. Okay. Now consider, here you will see the sarcotesta. The 2 layers of the sarcotesta are there, and then there are the 2 layers of the sclerotesta. Okay. Here are the 2 layers of the sclerotesta and 1 layer of the endotesta. So 2 layers of the sarcotesta, 2 layers of the sclerotesta, and 1 layer of the endotesta. So 2 plus 2 plus 1, that is the 5 layers, can also be seen in this probable fossilar seed. Okay. Now see the second family, which is called as the Porous Hyalure family. We have studied the Cordaitales family. Now here we have the second family, which is called as the Porous Hyalure family. See, it is the unigeneric family. That literally means it contains the one genera family represented only by the Porous Hyalure, a genus of the Permiocarboniferous age. Okay, Porous Hyalure is the genus which comes under the Porous Hyalure, and this genus is showing the presence of different kinds of fossils. It has the combination of the Pteridospermian and the Cordiaitalian characters. That is, the Pteridospermian characters are there, and the Cordiaitalian characters were also present in this Porous Hyalure family. Now consider this: the stem structure of the Porous Hyalure is resembles with the Gymnosperms of the Pteridospermials. Okay, during the Pteridospermials we have studied the Gymnosperms, and Gymnosperms like a stem, it's shown by the Porous Hyalure. Okay, whatever the Porous Hyalures are there under the family Porous Hyalure, and under this Porous Hyalure family comes under the Cordiatales order. This Cordiatales order contains the Porous Hyalure family, and this Porous Hyalure family shows the presence of Porous Hyalure genus. So the stem of this Porous Hyalure genus is similar with that of the Gymnosperms of the order Pteridospermials. Okay, the secondary wood in the stem of the Porous Hyalure was ill developed, and its medullary rays were much as the 3 walls wide. Okay, so secondary wood is not very much developed. This separates this particular family, which is called as Porous Hyalure, with that of the Cordaitales. Okay, in the Cordaitales family we have studied that there is a secondary growth, but here the secondary growth is ill developed. Okay, not very well developed, and this shows the presence of the medullary rays, which are nearly 3 cells wide. These are considered as a medullary rays today, and these medullary rays were 2 to 3 cells thick in the stems of the Porous Hyalure. Okay, now consider this. Over the dozens of the primary strands were present in a close contact in the secondary wood. Okay, whenever the secondary woods in the some form, whenever the secondary wood is developed, they were forming the close contact primary strands like this, which are comes in a very close contact with each other. Whenever the primary strands are come in contact with each other and they were showing the secondary growth, they create the structure like this, and it is considered then as a secondary wood. Then a large pith is there, which is surrounded by the ring of the exarch primary strand. So anatomically, although we do not know much about it, but the stem is showing the exarch primary strand, and this exarch primary strand in the stem is surrounded with the large central pith. Right? It is possessing the large central pith, and there may be ill developed secondary wood, or there may be the development of the secondary wood in the family of the Porous Hyalure. Okay, so this is all about the members of the Cordiaitales and the Porous Hyalure. All these 2 families were comes under the Cordiatales order. Okay, so the seeds which are reported in these family were termed as the Rudospermum, and were similar with the Cardiocarpus of the Cordaitales. That is, we have studied the seed of the Cardiocarpus in the family Cordaitales, and this Rudospermum seed which is found under the Porous Hyalure family, it is quite similar with that of the Cardiocarpus of the Cordaitales family. So we don't have to separately study the seeds of the Rudospermum. Okay, because the characteristics of the both the families seed are quite similar. Okay, whatever the characteristics of the seed possessed by the Cardiocarpus, it is also possessed by the Rudospermum as well. All right, so this is all about this topic. Okay, thank you very much for watching this video. Please like this video, share this video with your friends who are studying the botany, and if you haven't subscribed to the Botany channel yet, please subscribe to the Botany channel for UPSC examination. Again, thank you very much for giving your time. See you in the next video lecture. Thank you.